Humoral immunity relates to antibodies. We said that there are two main types of cells, lymphocytes T and lymphocytes B. Lymphocytes T are mainly related to the cellular immunity. Lymphocytes B are related to the humoral immunity. The main reason for that is because the lymphocytes B are basically the ones that are going to be responsible for the production of antibodies. Recognition. So we have the antigens right here. The lymphocyte B is going to phagocytize these antigens, and then is going to process that, and then the epitopes of these antigens are going to be displayed right here in the major histocompatibility complex. Number 2, antigen presentation. The lymphocyte B is going to process that antigen and is going to display the epitope right here in the cell membrane using the major histocompatibility complex. Then, once this is available in the lymphocyte B cell, the T helper comes over here, and the T helper is going to identify the antigen being displayed by the lymphocyte B. What the T helper is going to do is going to release interleukins. What interleukins are going to do are going to stimulate the production of more lymphocytes B. What is that called? Clonal selection. What happens with these lymphocytes B? They're going to go through a process of differentiation. After differentiation, these lymphocytes B will turn into plasma cells, and some of them will go into memory B cells. The plasma cells will produce the antibody. The response that we call it attack. So let's keep scrolling down. So you have in here two pictures, one of them showing you the B cell, and in here you have the plasma cell that is obviously larger, right? It has a whole bunch of Golgi apparatus in here. So right here we have the antibody. Skip this part for 1 second, and let's take a look at how the antibodies are going to stop the antigens, or how they're going to make them not responsive or not being able to initiate an immune response. So there are basically 4 mechanisms. The first one is called neutralization. In this case, the antibodies are going to neutralize the antigen by covering it. Since it covers it, then obviously the antigen will not be able to trigger an immune response. Next one is the complement fixation. Complement, remember, it got to do with the complement mechanism that we covered before. So in this case, IgM and IgG are going to attach to the antigen, therefore initiating the complement mechanism that eventually will finalize with the inflammation, phagocytosis, immune clearance, and cytolysis, which are the results of the complement activation. The next one is agglutination. Agglutination is the ability that antibodies have to attach to multiple antigens. Then they are able to bind them together, therefore rendering them inactive. Agglutination is what happens with blood type. You have an example in here, antibody attaching to multiple red blood cells. Why? Because if the antibody has multiple areas of attachment, precipitation, antibodies link multiple antigens, and they form these big antigen-antibody complexes. They are so big that they cannot remain dissolved in the solution, and because of that they precipitate into different areas of your body, and they can be removed by macrophages, for example. So if we scroll down, we have 5 types of antibodies: AGA, AGD, AGE, AGG, AGM. So in the case, AGM plays an important role in precipitation and agglutination because it has multiple areas where it can attach to multiple different types of antigens. IgM represents 10% of the antibodies present in plasma. Next one, we have IgG, one of the antibodies that is going to be secreted in the secondary immune response. It's also very important in complement fixation. IgG also is able to cross placenta, therefore able to provide some immunity to the baby. IgE is related to eosinophils. IgE is commonly found in inflammatory as well as an allergic reactions, as you can see right here. IgD helps with the activation of the B cells. IgA is very common in tears, saliva, mucosa. So let's take a look at the antibodies. Right here you have the characteristics of the antibodies. It's going to have heavy chain because it's long, light chain because it's short. This area right here is the antigen binding site. This is the area that is going to change, therefore it's going to be called a variable region. This area and this area don't change, therefore they are called constant regions. They are attached to each other right here using disulfide bond. After all this process happens with the antibodies, we are going to get memory. The memory obviously is very important because you're going to have antibodies that are going to play an important role in it. You have in here primary response. You have the infection for the very first time with an antigen. Right here you can see in day 5, basically you're going to have IgM that happens first, and then IgG. This initiates the memory. What happens if you get another infection with the same type of microorganism? In that case, you're going to see here the secondary response right here. There's no delay in the presence of IgG and IgM. It's an immediate response, therefore not allowing the antigen to cause any sort of problem in our body. And as a matter of fact, you can see how high and how strong the response of our body is with the production of antibodies in order to destroy the microorganism.